throughout development. It's probably not the same across species. However, within nematology, you'll oftentimes see statements in textbooks that the neuroanatomy of nematodes is highly conserved. Okay? And you'll see this very frequently. And it's basically based on an N of 2. Okay? So it's based on this gigantic beast, Ascaris suum. So this is a gastrointestinal parasite. This one is particularly found in pigs. And then to put it to scale, here's C. elegans down here. So they have almost the exact same number of neurons. The connectivity of them is very, very similar. And so based on that, these data, you think, okay, these two nematodes are totally different. The nervous system, though, is remarkably the same. It must be true for all nematodes, right? That they all have a conserved nervous system. So as a postdoc, as a sort of a side project, and then when I started my own lab at Illinois, I had my first grad student basically do more of a systematic study of nematode nervous systems. And one of the structures that I'll talk about more today is the ventral nerve cord. So this is right along the ventral edge of the nematode, and it's basically a series of motor neurons. So motor neurons are neurons that directly connect to muscle and basically tell the muscle, okay, contract or relax. Okay. In C. elegans, as I mentioned before, the entire nervous system was completely reconstructed from EM. We know every single neuron in that ventral nerve cord. If we do DAPI staining, which is just staining the nuclei, we can go through each of those DAPI stained nuclei and say, okay, I know that this particular neuron is AS7. Okay. Part of the reason we can do that is that the nuclear morphology differs between cell types. So neurons have a different nuclear morphology than intestinal cells, which is different from muscle cells, which is different from epidermal cells. Okay? So what I asked my first grad student to do is to do a comparative approach. And so I'll just show you one piece of data from this. So he compared the ventral nerve cord across a bunch of different clades. This is an incomplete listing of some of the species he looked at, and found that there are substantial differences. So C. elegans has 57 ventral nerve cord nuclei between these two structures called the retrovesicular ganglion and the preanal ganglion. Don't worry about remembering that. And Ascaris has 55. Okay? But what you find is that there's a fair amount of variability across these clades. Okay? He then went on and looked at a set of sensory neurons, so neurons in the head that we were able to stain with lipophilic dyes, and found variability there. He also found variations in the timing of development. So C. elegans has a very, very specific timing when the ventral nerve cord develops. And what he found was that also was not conserved across species. So it makes sense. You wouldn't expect it to be conserved, but the dogma had been that it was highly conserved. So this same student, Paul, after he got through this project, he really became more interested in sort of how the nervous system functions, okay, rather than taking a descriptive approach. And so because of that, we were toying about with different ideas, and we sort of settled on, let's try and look at some neurotransmitters. So neurotransmitters are basically small chemicals, small molecules that signal from one cell to the other, from a neuron to another neuron, or from a neuron to a muscle, and tell that downstream cell to do something. Okay? One neurotransmitter that many of you have probably heard of is serotonin, structure shown here. Previous work in the C. elegans field, as well as in plant parasitic nematodes, showed that if you take exogenous serotonin you just get from sigma, you dump it on the nematodes. In C. elegans, which is shown up here, the entire esophagus will start pumping. Okay? Similarly, if you dump serotonin on plant parasitic nematodes, so this is actually SCN, it's molting from a J2 to a J3 stage, the stylet, which is the sort of feeding apparatus, it's a spear right at the tip of the head that it used to break into cells, this will start thrusting out. Okay, and I'll show you a video of that in a second. So in the C. elegans field, they went on beyond just this exogenous serotonin to find out, okay, how does endogenous serotonin actually act? What are the genes involved? where is the serotonin being expressed. With the plant parasitic nematodes, nobody had really gone on to look at what was happening internal. Was there actually endogenous serotonin? We would assume so because it's a very conserved neurotransmitter. But that was sort of the project that I gave, second project that I gave to Paul was, is endogenous serotonin playing a role in plant parasitic nematodes? So the first thing that he did, first I should say that instead of looking at SCN, we looked at a different nematode called Pratolincus penetrans. This is a lesion nematode, so it has a very wide host range, very commonly found throughout the Midwest. And he did sort of a study that other people, including myself, had done, but actually had not been done in root lesion nematodes. So this was a case where I said, well, I'm not sure if any of the endogenous stuff will work, so let's try Pratolincus because nobody's even thrown serotonin on it. So he put serotonin on it, and he found that compared to the control, you got an increase in the number of stylet thrusts per minute. So we wanted to see, okay, is there actually endogenous serotonin within lesion nematode? So he did a couple of things to look at this. So the first was antibody staining. So there are some good anti-serotonin antibodies. So he did antibody staining, and he found right around the esophagus of Pratolincus, he found a couple of cells that really stained brightly for serotonin. Interestingly, he didn't see anti-serotonergic cells within the esophagus itself very consistently. But it did suggest that serotonin was being produced. The second strategy he took to look for endogenous serotonin was a pharmacological approach. So what he used, so here are the serotonin results as before. So the black bar is the tested chemical versus the white bar, which is control, and looking at stylet thrusts. What he did was he used a couple of serotonin reuptake inhibitors. Okay, so if we think about neurotransmission, so if this was the cell that's releasing serotonin, it's going to sprinkle out its serotonin here, and then there's going to be a receiving cell that's going to receive the serotonin molecules and going to be activated. Okay, well, as animals, we don't want that serotonin just hanging out there and constantly hitting that receptive cell. Otherwise, all of our bodies would be going crazy all the time. So one of the tricks that animals use is on this cell that's sprinkling the serotonin out, they also have other proteins that are basically sucking it back in so that there's sort of a controlled amount of serotonin that's being released. There are pharmacological compounds that can basically block that reuptake. Fluoxetine and imipramine are one of these. Many of you probably heard of the common name of fluoxetine as Prozac. Okay, so basically we took our nematodes, we put them on antidepressants, and we saw, okay, what was the effect? Okay, so the idea is, if there's endogenous serotonin that's being released, and we block that reuptake, it should basically act the same as putting a bunch of extra serotonin on there. If endogenous serotonin is not being produced, well, then those blockers of the reuptake should have no effect. But what he found indeed was that both fluoxetine and imipramine had similar effects to the serotonin. He then went on and looked at other behavioral aspects, so he looked at male-specific structures as well as the egg-laying apparatus of the females, and found that there are serotonergic cells there, and that these compounds would also affect those behaviors. Okay, so those were sort of the appetizers. Hopefully I've whetted your appetite for thinking about nematodes and neurobiology. And now I'll get on to the main course. And again, so these are unpublished data. We're hopefully, as I'm speaking, collecting data. I don't know what they're doing in the lab right now. But this is going to focus on the soybean cyst nematode Heterodera glycides. Okay. Now, since this is a plant pathology